We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. CARTA brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
So this is my show and tell. Every single cell in our body is packaged with my toolkit, which is DNA. And we have two types of DNA that carries the blueprint that we all walk with. And I know to this audience this is like kindergarten school, but I'm showing this to remind us that I'm going to talk about two different types of inheritance. The nuclear DNA, which is makes up the chromosomes that undergoes mixing at cell division or recombination. That is now why I am at this height and at this size. You heard about brain size and height and weight, etc. Well, I am your show and tell for all of that today. In addition, small is packaged and dangerous. We have the mitochondrion that has its own unique set of DNA, which over evolution has come to carry yet another remnant of DNA in every cell. Now, the reason why the mitochondrial DNA is of use is that it is strictly maternally inherited and carries a trajectory of our maternal inheritance. So these are my two show and tell sources of information, and so that I don't exclude the males in the audience, yes, you have that little thing there, the Y chromosome. Well, genomic inheritance is known to all of us. We get half our chromosomes from each of our parents, and they from their parents before, and so on, and so on, and so on. And so with respect to mitochondrial DNA, we would be able to track what both sons and daughters got from mother, from maternal grandmother, and great-grandmother, and so on, and so on, and so on. In males who have the Y chromosome, we have the equivalent mechanism to trace paternal heritage. But of course, the nuclear DNA carries the contributions from both parental sides. And if you go back 1 to 3 generations, you have 8 people who potentially contribute to your ancestry. And if you want to go back 20 generations, 2 to the power 20 would have contributed to your total genomic information, but you'll still have a single maternal ancestor. And it was just by chance that I chose for my title, So Where Do We Come From?, not knowing that that is among the questions that CARTA looks at. And the reason I chose that was about 15 years ago, I was involved in a documentary that was produced in South Africa by one of the TV networks by the same name. And in that we showed a whole bunch of local people transcending politics, sport, and all sorts of disciplines. And we did their genetic ancestry tests and made a story out of it. And I was going to show you some clips from that, but it would have eaten into my limited time that I had. But in any event, you've heard about fossils, you've heard about tools, you've seen a little bit about culture, and basically those subdisciplines allow us to track from the past and build up to the present. The genetic toolkit is the reverse. We study the DNA found in living people, and we reconstruct the changes over time. And for this demonstration, I'm using mitochondrial DNA, which is only transmitted through the mother line to daughters, and only daughters will pass it on. And the sexy thing about this DNA is that it does not shuffle or recombine. So you have a mechanism and a valuable tool to reconstruct the history of change over time to some point back in the past. And if I were to demonstrate this with a little bit more of a cartoon, so if we were to take mitochondrial DNA, we'd be able to link it to some point back in the past. We call that the most recent common ancestor for mitochondrial DNA. And the reason why I dislike the use of the terminology mitochondrial Eve, it gives a sense that the most recent common ancestor was the only female present in that time zone, which is not the case. So, but in reconstructing the lineages found in living people, the divergent lines kind of coalesce or come together at a common point, some in the distant past, dated to between 150 and 200,000 years ago. Now, mitochondrial DNA was made famous by the seminal paper by Becky Kahn, Mark Stoneking, and Alan Wilson, where they used mitochondrial DNA. And in the 80s, we didn't have the sophisticated technology of polymerase chain reaction to make lots of copies of DNA from small amounts. These authors went out, collected placenta, which was a rich source of DNA, extracted the DNA, cut it up with little protein tools called restriction enzymes, and created these maps. And what they found was, when they looked at the relationships of all the different patterns that made up the lineages, one particular branch was exclusively containing individuals from Africa. And as you went towards the rest of the tree, drawn here as the famous horseshoe-shaped tree, one found that in the shallow parts of the tree, individuals from Europe and Asia were positioned. So this kind of spearheaded the out-of-Africa theory concerning modern human origins. It wasn't without controversy. There were many, many side chirps around this, criticizing the technique for the dating methodology and so on. But that's a long story. But several other types of studies started to come to the fore. And what was now becoming more and more apparent was that as you went further away from Africa, and for our colleagues from East Africa, that's seen as a center of the evolution of our species, as you went further away from Africa, the amount of genetic diversity was becoming more and more reduced. And this was further evidence that our species originated in Africa. I don't think the world was still ready for this because, just think about it, all the social issues going on about coming from Africa, and that's like another 5 lectures to go, but I will spare you that. Nonetheless, as the technology started to improve and we've gone from restriction mapping to the advent of the introduction of polymerase chain reaction, better techniques for genetic sequencing, and now we can do whole genome sequencing. The culmination of the Human Genome Project gave us scientists, an open era of asking questions and trying to resolve that through analysis, etc. And so some colleagues sequenced a few individuals from their whole genome sequences from Southern Africa. It was the first sequences from the Southern African region, and included among those individuals, there were 4 people from Namibia who were San, and our famous Archbishop Desmond Tutu. And so when we got the results from there, all individuals, with respect to their mitochondrial DNA, had similar patterns to that of the San and the Khoi, the indigenous people from Southern Africa. And that is what made the news. Even Oprah Winfrey's show and all the news bulletins were talking about Archbishop Desmond Tutu being part and parcel of ancestry to the first people. This was a whole genome sequence. The mitochondria is yet but a small component of it, but the world was talking about the mitochondrial genetic study showing linkage of Archbishop Desmond Tutu to that of the San. Now, what the study brought out was quite an interesting observation. Using the x-axis in a plot of summarizing all of the data collectively, each individual in a pairwise fashion, you saw that there was one cluster that was associated with African people from the Niger-Congo language family, and there was another cluster that contained those 4 San individuals, and those people from the European regions, can you see it? Just clustered into like one spot. So immediately you can see, just with a few sequences, the vastness and the diversity present in Africa. And if you were wanting a magnified view of that, if you just looked at the African individual, once again, you saw this increased diversity. But there's something emerging from here, that people from different parts of the continent, their genetic structure was seemingly different. And, we had theories about multi-regional theory concerning all human origins, and people started to talk about asking the question about evolution in Africa. And so already we're seeing there's specific patterns in the southern part, in the western part, and in people who were migrant to the southern part, similar to that in the wider part of Africa, namely people who are speaking Bantu languages. And so my group has been for over 3 decades now. I have been in this business from the very beginnings when DNA technology kind of hit the world, and we picked it up in South Africa almost like a backlash 10 years later. And so I came into the business when we were still using very simple techniques to the point of where you can do whole genome sequencing now. And over the years, through my predecessor, Professor Trevor Jenkins, we've been interested in the history of peoples of Africa, and we have always tried to do it in a holistic way, using genetics as our tool, but also engaging with colleagues from other disciplines, linguistics, archaeology, paleontology, history, and anthropology. And so I'm a bit of an apprentice of all of that and consider myself more a molecular anthropologist than a hardcore geneticist, because I'm interested in these sorts of questions. In any event, I had a very bright PhD student, Karina Schlibusch, who was interested in following up on some of the studies we were doing among the Khoi and the San populations. She's now doing amazing work with Matthias Jacobsen in Sweden, and last week I had the privilege of being at a meeting there in Sweden where they brought together the world's best working in the disciplines of linguistics, archaeology, and genetics to talk about the diversity in Africa. And while we present a very simplistic overview here, the more recent work is actually very dynamic. There are lots and lots of debates, and the thing that was most mind-boggling was that there are researchers in France now modeling linguistics, archaeology, and genetic data collectively to kind of get a deeper vision of the past. In any event, we did some SNP chip technology, moving away from just mitochondria and Y chromosome that we'd been busy with for a while. And what we found was that the first branch in the human tree with this kind of genomic data contained populations that were exclusively Khoi and San speaking peoples. They had genetic information greater than 100,000 years. And the first time there was a divergence from that major branch was as recent as 45,000 years ago, where we see some splits in populations from Central Africa, Eastern Africa, and West Africa. So that was quite a major observation. We were not the only ones who made that observation. Others have done so as well. Now, this is a scary slide, but just allow me to walk you through it. This is another analysis that people do with genomic data. It's called structure. And basically what they're doing is looking at each individual. So if we have 500 individuals in our sample, each individual is a data point across from left to right, belonging to that branch where their sample is placed, and its amount of admixture. Okay? So there are no pure populations. Let's just get that first fact out of the way. There are no pure populations. People seem to think when you do admixture mapping, you're starting with 2 pure populations and then you're blending. No, we are mongrelized ahead of that. And so now when you look across using structure, it identifies these different genetic backgrounds. And all I want for you to see that if you go with the green being West Africa, the blue Central Africa, and the red, the Southern African Khoi and San, if you look from left to right, you can see different color structural patterns. And so this is what is going on in Africa. There's a deep structure of genetic patterns of difference across the populations on the African continent. And if I were to just summarize that by a very, very nice review written by Karina Schlebusch and Matthias Jacobsen published last year, then you would see a summary of what we've seen before with a difference. What has now happened is by looking at some ancient DNA samples, both in South Africa and in Central Africa, in studies published by different authors, they've now pushed that convergence point of dating the common ancestry in Africa beyond 150 to around 300,000. And that is in keeping with some of the fossil evidence that's come from Flores Bud as well. So when you now look at that genetic pattern I presented earlier and you condense it all together, we see once again on one branch the Khoi and the San populations, Central Africans, West Africans, and East Africans. And a small branch out of that represents what's come out of Africa. So you can see now why Africa is every expeditioner's paradise. Because if you want to do biomedical research, and if you do not understand what's come out of Africa, you might as well pack your bags and go to the beach, because this is what you need to understand. And so we feel very privileged to be in that region, but it is also quite difficult, because while I may sound very enthusiastic about it, of course, we have to also work with all the ethical, legal, and social issues that come with working with human subjects. Now, if I were to just very quickly summarize graphically, in keeping with a map of where those genetic patterns are seen, you see we also have many gaps. So scientists are busy collecting samples to try to fill up those gaps, and the data is out there, it's just coming into the public domain now. So what we see in Africa is very deep history. What my colleagues ahead of me have been talking about is that very, very deep history. But how far back do we take the genetics in Africa is unlike in Europe, where we've got samples from Neanderthals from 45,000 years ago. If some of the archaeologists and paleontologists will allow the scientists to touch their specimens from Africa, maybe we could go deeper. We did publish a paper 2 years ago about specimens dating to just over 2,000 years ago to try to understand what would have been the genetic structure of individuals pre the migration of people speaking Bantu languages to the south. And that paper was published, and I didn't want to bring it here because that would have taken 20 minutes to explain. So we have very deep structure. We have migrations associated with the spread of Bantu languages from West Africa into Southern Africa in the time period spanning about 3,000 to 5,000 years. And then we have a very interesting pattern with the advent of pastoralism. As people started to acquire cattle and move to the south, the Khoi, who we've sampled in Namibia, carry a genetic pattern linked with lactase persistence, the ability to consume cow milk, similar to that found in the Maasai in Kenya. So that's another very, very interesting adaptive trait that has come into the southern part of Africa. Now, for those of you who've been following this literature, you know that people in Europe have different genetic markers that allow them to process to consume milk. But in Africa, different genes. So to the same cultural trait, different genes have been adapted for. And so in addition to that sort of tracking of mobility, we've also had input from Europe and Asia into the African continent at different times. So not only did Africa give the rest of the world its genetics, those individuals who were in exile, some have returned, like me. So a couple years ago, as I started to engage with colleagues outside of my discipline to make sense of genetics, I would go to archaeological meetings and, engage with other colleagues. And it became very apparent that even among our academics, we were talking across each other. I mean, I would get people calling me up and say, well, if you were to go to a particular gravesite where skeletal remains were found, can you tell us if those were slaves brought from Zimbabwe buried there? And so I would ask, well, did you think that Zimbabweans had a different genetic pattern to other Africans? So we were talking across, understanding each other. And so I was privileged to have hosted a conference in South Africa where we invited multidisciplinary colleagues from within the country and overseas to participate, and I was able to edit a book, one of the first on the prehistory of Africa, where it was a collection of bringing together this kind of interdisciplinary dialogue. And we've progressed to being able to talk a little bit more now, so much so that all of our genetic studies has at least an anthropologist or a historian or an anthropologist associated with it. Colleagues, the biggest story is about translation of science. When we sit in our little ivory towers and we communicate just with our peers, we forget that the individuals that we need to serve are the general public. And so in my own field work, having to work outside of the ivory towers of university boundaries, you really, really get acculturated into what society is all about. And fortunately for me, there's always a tree somewhere, and I would use the metaphor of a tree to talk about all living people being a leaf on the tree, and your connection through branches to the common trunk is actually the history of how your DNA links to the common trunk. It has the opportunity to bring people together, to realize that we are one species, bringing human solidarity into the dialogue, and also having the ability to tell stories. And so I created, with a company called Jive Media in South Africa, some of these tools where we're telling stories. And as you tell the story, here the grandmother, the gogo, is telling her family how her people have come to be where they are. That's oral history. Cultural anthropologists collect information on oral history. And we intertwine the story into this cartoon where one of the individuals has been to university and he's had a genetic ancestry test. And so the young buck, the curious little guy, wants to know all about it. And so in unfolding the story with different cartoons, we now unpack what a genetic ancestry test is. Now, because that is what I was interested in, we use it as an opportunity because lots and lots of people are doing biomedical research in the country. And that whole issue of informed consent is not only about the signature, that informed part is actually very powerful. And so we try to have tools to be able to take the jargon out of the conversation and bring in a visual to help the process. So I just wanted to put that little punt in and to say that the science that we do could not stay out of the public domain. It very quickly crept in with opportunities to engage the public. People demanded to have genetic ancestry tests. And so we had to set it up, and part of that documentary, I even was privileged to have had the opportunity of testing former President Nelson Mandela. And his mitochondrial DNA was present on this branch. So this is my famous tree, which is actually adapted from the tree that was published by Behar et al. In a schematic way so that we could show how the different branches are related. All the branches with green circles carry mitochondrial lineages commonly found in Africa. And then as you come out of Africa, the M branch is seen in the Indian subcontinent and into the Americas. And through the N, we see most of the branches that are found in Europe and some in Asia called Eurasian lineages. So this is a summary of the mitochondrial DNA patterns that exist in living humans today. So former President Nelson Mandela's lineage was found on this branch L0, which is the one found most commonly among Khoi and San. And believe it or not, having now done research in various communities, one of the things I've tried to do is take back a result, a mitochondria and Y chromosome result, to individuals so that they don't feel that parachute type of science that, you go in, you take the samples and you go out and never to be seen again. So we go back, giving them those results. And so now some other unique thing has come about. People are now calling me up almost on a daily basis to have their genetic ancestry test done, because in some areas, they've been told that if you have your genetic test, it will be your certificate to tell you whether you are San or not. And in that way, you may be able to claim land in the whole land claim issue that's going on. So what started off as a very complementary way of keeping the relationship going between the science and the communities, it's now coming back where there's opportunistic aspects being brought into it. So one of the second fact, the first one was that we are not pure populations. The second fact that you must go away with is that a genetic ancestry test does not tell you anything about your identity. As you sit here, you have myriads of identities, huh? But no genetic test could define any one of those. So that's the second thing I like to emphasize to people, that genetic ancestry is not equal to identity. Identity is complex, it's multifaceted, and it's fluid. Your genetic ancestry is the unbiased record of your genetic heritage going back in time. So colleagues, genetics, that was just genetics 101 or genetics 0.05, has been a very, very useful tool to try to track movements of people around the globe. Not only have we used mitochondrial DNA and Y chromosome DNA to kind of plot the phylogeography of the human journey, the advent of whole genome sequencing and various types of analysis with that data now is refining theories concerning our origins. Ancient DNA has allowed a deeper view into the past, and I don't have time to talk about those components, but it is another way of gaining insights into questions that we would otherwise not have had opportunities for. So in closing, I first of all want to acknowledge with deep gratitude the CARTA Institute for this opportunity to be here, participants who obviously make us who we are in our careers, various collaborators and funders, and I have to give a punt wearing the cap of my new role as the Executive Officer of the Academy of Science, is that science should be done to promote and advance science, evidence-based science in serving society. Then there's science for society, and of course, science for policy. So the next time you venture into your science world, just try to deposit a few kudos in each of these 3 boxes. Thank you very much.